of a life cycle. Butterflies go through some fairly spectacular life cycle transition, turning from something that looks like a worm into a pupa, and finally into a glorious creature that floats on the breeze. Other organisms from humans to plants to bacteria also have a life cycle, a series of developmental steps that an individual goes through from the time it is born until the time it is produces. Question is, what is the eukaryotic cell cycle? The eukaryotic cell cycle is an evolutionary conserved process that results in the replications of the cell. Stages of the cell cycle. To divide, a cell must complete several important tasks. It must grow, copy its genetic material, and physically split into two daughter cells. Cells perform this task in an organized, predictable series of steps that make up the cell cycle. In eukaryotic cells, or cell with a nucleus, the stages of the cell cycle are divided into two major phases, the mitotic phases and the interphase. But today, we're going to talk about the interphase. During interphase, the cell grows and makes copy of its DNA.
Preparation for division happens in three steps. First is G1 phase. During G1 phase, also called the first gap phase, the cell grows physically larger, copies organelles, and makes the molecular building blocks it will need in later steps. The second is S phase. In S phase, the cell synthesizes a complete copy of the DNA in its nucleus. It also duplicates a microtubule organizing structure called the centrosome. The centrosome helps separate DNA during M phase. Lastly, G2 phase. During the second gap phase, cell grows more, makes proteins and organelles, and begins to reorganize its content in preparation for mitosis. G2 phase ends when mitosis begins.
The G1 checkpoint is the main decision point for a cell in which it must choose whether or not to divide. Once the cell passes the G1 checkpoint and enters S phase, it becomes irreversibly committed to division. That is, barring unexpected problems such as DNA damage or replication errors. A cell that passes the G1 checkpoint will continue the rest of the way through the cell cycle and produce two daughter cells. To make sure that the cell division goes smoothly, the cell has an additional checkpoint before M phase called the G2 checkpoint. At this stage, the cell will check for DNA damage and DNA replication completeness. If errors or damage are detected, the cell will pause at the G2 checkpoints to allow for repairs. If the checkpoint mechanisms detect problem with the DNA and the cell cycle is halted, and the cell attempts to either complete DNA replication or repair the damaged DNA. If the damage is irreparable, the cell may undergo apoptosis or programmed cell death. This self-destruction mechanism ensures that damaged DNA is not passed on to the daughter cells and is important in preventing cancer. The M checkpoint is also known as the spindle checkpoint. Here, the cell examines whether all the sister chromatids are correctly attached to the spindle microtubules. Because the separation of sister chromatids during anaphase is an irreversible step, the cycle will not proceed until all the chromosomes are firmly attached to at least two spindle fibers from opposite poles of the cell. The question is, how do the checkpoints actually work? Internal and external cues trigger signaling pathways inside the cell that activate or inactivate a set of core proteins that move the cell cycle forward. That concludes our overall presentation for cell